Answering MCAT questions in under 60 seconds. A researcher is investigating metabolic differences in patients with a rare metabolic disorder that results in chronically elevated glucagon levels. Following a standardized high-carbohydrate meal, which of the following best predicts the effect of this hormonal imbalance on lipid metabolism in these patients compared to healthy individuals, right? So remember that glucagon is going to be a hormone that sort of promotes breakdown of energy storage molecules, right? Like glycogen, like triglycerides, right? And then into their monomers, and then promotes the release of those monomers into the bloodstream, right, to raise glucose blood levels, to raise lipid blood levels, right? And it also promotes things like gluconeogenesis to raise glucose blood levels, while at the same time inhibiting processes like glycolysis, like fatty acid synthesis, protein synthesis, etc. Okay, so here we would expect exacerbation of those effects since glucagon levels are abnormally high. So let's see. A says reduced de novo fatty acid synthesis in the liver due to decreased activity of acetyl-CoA carboxylase. Well, we would expect glucagon to reduce fatty acid synthesis, right? Because it's trying to do the opposite of that. It's trying to cause fatty acid breakdown, right? So A actually sounds like it would be an expected effect, right? B says increased triacylglycerol storage in adipose tissue due to enhanced glucose uptake, right? Well, glucagon certainly isn't going to facilitate glucose uptake. It's actually going to facilitate glucose removal from cells and entry into the bloodstream to raise levels of glucose, right? So that would be more of an effect of insulin, not glucagon, right? So B is wrong. C, elevated lipoprotein lipase activity in muscle tissue resulting in greater fatty acid deposition. Well, this is suggesting basically an effect that's going to promote uptake of fatty acids by cells and removal from the blood, not the other way around, which is what we would expect glucagon to do, right? So this would be more of an insulin effect. So again, wrong. And then D, increased conversion of citrate to acetyl-CoA in the cytosol, promoting fatty acid synthesis. Well, again, we would be expecting less fatty acid synthesis with high glucagon, not more. So D is wrong too, and A is the best answer.